Okay, let's get underway. So it's my real pleasure to welcome Professor Richard Hellar here today. Richard is truly a man after my own heart. He discovers basic stuff, but he's also interested in taking those ideas and trying to have an impact on the wider world. But he also has that kind of, he's interested in tinkering with things and machines and making things work. It's like you're a throwback from the 19th century, Richard. You're wrong time.
Could be.
Anyway, he's been doing some wonderful stuff. He had he had a history-making deal recently. His treatment for melanoma involving electroporation of IL-12 into tumors in association with checkpoint inhibitors just triggered, I think, the largest venture deal. It wasn't?
I wasn't part of that one.
You weren't part of that one. Okay. I've been correct.
It was our other technology.
The other technology.
Yeah.
Okay. Well, anyway, it's the sort of thing that gets venture capitalists very interested. And his phase 2 clinical trial work on this melanoma treatment has been very promising, and it looks like it could be a real step ahead into treatment of this awful disease. So we're going to learn today. I saw a picture of a heart flash up there briefly. So it's not just skin; it's heart as well. So anyway, I'm looking forward to learning what you're going to teach us. Briefly, Professor Hellar is Professor and Eminent Scholar of the Frank Reidy Research Center for Bioelectrics at the School of Medical Diagnostics and Translational Sciences, Old Dominion University in Norfolk. So the floor is yours, Richard.
All right. Thanks, Rob, for a really great introduction. Thank you also for the invitation to share some of our results. So hopefully I'll live up to this introduction and you'll see some nice stuff. So, so what we sort of focus on is figuring out how to effectively do gene therapy or gene transfer. And as you know, gene therapy is being tried now for 2 to 3 decades, and there have been some good advances. Recently, there have been some approvals, both in Europe, and we had the first approval in the United States just recently for AAV to a rare inherited eye disease, which actually is one of the big breakthroughs. And as you can imagine, there's lots of challenges to this. Of course, the first thing is to figure out what exactly are you going to be targeting, what's the application, and the most obvious one, of course, is protein replacement or correcting a genetic disorder, which is very difficult because you have to target every cell that has that defect. But there's also vaccines and immune stimulation as well as some others. And how you might do this is you first have to figure out what, of course, is your transgene. What are you going to replace? What are you going to use for that? And then how you're going to target it. And one of the biggest stumbling blocks, which is what we're kind of focused on, is how do you deliver it? And there's 2 basic categories, very imaginative, biological and non-biological, right? And if you think about the biological, it's going to be viral or bacterial vectors, and non-biological is plasmid DNA. There are pluses and minuses to both. I won't really get into that. I'll tell you my biased opinion that plasmid DNA is better, but mainly because it's safer, it's easier to control. And one of the interesting things that you might not think about is that with plasmid DNA, you could actually get at the lower levels. And most people think, well, you want to get in there and you want to get high expression levels, but that's not always the case. And I'm going to show you an example where it's not the case. And then, of course, safety is always a big issue, especially when you want to translate to humans. With viruses, you have to worry about responses to the vector. You have to worry about integration and toxicity. Not so much with plasmid DNA. You kind of eliminate a lot of this. And then the key is going to be control of that expression. How long does it express? How much expression is going to be there? Some of that can be controlled by the construct. You could have an inducible promoter, which can work to some extent. And then interesting, in Europe, they actually do not like the idea of an antibiotic resistance gene being incorporated into these plasmids, especially that are going into humans. And so you have to come up with different ways of constructing them without using that. So the other thing when you think about it, when you think about standard therapies, of course, you're thinking about recombinant protein and ways. And of course, this is, in a way you're doing this, it's a known concentration, you can control the dosing, but typically it's got a short half-life. And so, and because of that, usually you have to give it at high doses, and this could lead to toxicity. It also can be, even with giving at high doses, you could end up with low concentration where you need it. The other thing is that some recombinant proteins can induce an immune response, depending on the source of that protein. And this is because there could be isolated from human blood or tissue, and you may get a reaction to that. It's also possible to get reactions or infections when they're taken from animals, such as bovine sources. And then once you have them in there, the question is, can you downregulate the protein levels? And this becomes difficult. So even though the positives of understanding the pharmacokinetics and the pharmacodynamics, there's all these shortcomings of protein therapies. And if you think about it, this is where a lot of problems end up coming in how do you control that protein dose. With gene therapy, you can get continual production of that protein, right? So you don't have to keep infusing the patient. And it's produced by the patient's cells. So the modification of that protein is usually correct, and so you shouldn't get a response to that protein. You could also downregulate it using siRNA or shRNA, or as I mentioned before, inducible promoters. Then we got this issue with biological versus non-biological vectors. And of course, the big issue is that the protein dose is variable. And that's the big difference with here, where you know the dose and here you don't, mainly because what you're thinking about, what you're delivering, is based on the DNA concentration, not on the protein concentration. And we believe that we have figured out a way how we can better control that and understand what's going on. And that's using what we call gene electrotransfer. And it's a physical method. Essentially, we believe it's a safe alternative to other methods. What we do is essentially you're going to inject the DNA into the tissue. We place electrodes either into that tissue or on that tissue, apply some electrical energy, which causes permeabilization in the cells, and the DNA gets in, and hopefully it expresses. So why do I think this is a way to be able to control the dose? And this is a busy slide, and the reason why it's a busy slide is because it's a daunting task to do this. So, and that's the only reason why I kind of like to put this all on one slide. And basically, these are all the variables that you could control when you do electrotransfer. You know, the parameters for the actual pulse, how long the pulse is, how much amplitude the voltage is, how many pulses, direction of the pulses, and then also the selection of the tissue target. Each of these tissues, although you wouldn't think it, but because there's different cells within there, the expression profile is going to be different depending on which tissue you select, as well as your electrode configuration. So when you think about all these different things, as well as DNA concentration, you can manipulate all these different aspects to either get high or low expression, or short or long-term, or local or systemic expression. And it seems like it's very difficult to do this. And to some extent it is, but it ends up being very worthwhile doing it. And once you start knowing the basic premises of how this interaction occurs, it becomes easier and easier. And you can get to the point where you can predict what the expression is going to be. The other thing that you have to be careful of is that it is transgene dependent, not just tissue dependent. And there are some differences between the reporter versus the effector. So if you're doing a reporter gene to figure something out, you have to make sure that the gene that's going to give you the effect is going to have the same type of profile as the reporter. And it will be specific to the application. And we very strongly believe that the kinetics and the target is going to influence the therapeutic outcome. So this becomes very important to do if you want to be able to successfully translate and be able to get the therapeutic outcome that you want. Sure.
Question, Rich. Can you go back to that previous slide? So this is a comment, but I'm interested in your perspective. So when you look at all the aspects of the protocol you can control, which you put up there, I'm thinking about a parallel. We work in the nervous system to stimulate cells and axons that fire to create synaptic plasticity. And so one of the things that some of my postdocs or may or may not be in the audience play around with is the pattern of pulses as well. And so you can take a given frequency and a given duration, a given number of pulses, and modify the statistical distribution, what we call the regularity, the irregularity, coefficient variation. And in our case, in the nervous system, that matters. It can matter under certain circumstances, and it's accessing downstream signaling pathways differently.
Right.
So I'm just curious if, I'm thinking about how you can permeabilize things and open things up in the relaxation time, whether that parameter has ever been investigated in
Not to the extent that it can be, but people have looked at that. So they've actually, there has been some groups that have looked at actually using pulses that are typically used for muscle stimulation. Long trains of very, very short pulses to be able to deliver to. To some extent, what ends up happening is those end up being a little bit damaging on that. And on one side, and then the other side, they actually become very effective in delivering to an extent where they actually achieve some integration, which is kind of things that we try to avoid on that. However, I think I think you're right, that if you look at that in some ways, anything that can change the conductivity of the tissue, or in some respects, for lack of a better word, prime the cells to when you pulse them, can actually make this an easier protocol to do. So, and we've been actually looking at certain ways of being able to do that, not with necessary electricity, but with other things. But that might be something else to kind of look at. Yeah, it's a good point. Okay, just to kind of give you an idea, this is an old piece of data, but it's just really to make a point of the idea of what you're looking for. So it's really, this delivery is a threshold event, and it's a dual threshold event in that we have to hit at a certain threshold in order to get our delivery. So below this 1,000 volts per centimeter, in this case, this was 100 microsecond pulses. So if we used a different pulse width, this amplitude might end up being different. But in this case, it was 1,000 volts per centimeter. You can see we get nice expression. And we maintain it, and then all of a sudden it drops off. So at this point, we're starting to damage the cells. And the thing with delivering DNA, you have to realize that there's two things that have to happen. One, the DNA has to get in the cell. The cell needs to be alive because we want that cell now to produce the protein from that DNA, and you're using the cell's machinery to do that. So when you get to where you're damaging the cells, that's why your expression drops off. Okay, so I'm going to focus a bit on the skin and give you some ideas of how we're doing this. First, I'm going to talk about, just to give you kind of an example of how we go through the process to try and figure out how best to deliver to the skin. And then I'll show you a couple of applications where how this could work in a potential therapeutic application. And we picked the skin because, of course, you could do it in a non-invasive way, or at least a minimally invasive way, and it's easy to monitor, and we have lots of skin. I mean, skin is, some of us have too much skin, including myself, but it also makes it very easy to use. The other thing, especially for vaccines, is it has a lot of APCs, and so it's really good for stimulating antibody production. Okay, so just to give you an idea, these are a couple of standard electrodes, and they're very preliminary, very primary, or primitive. Primitive would be the P word I was looking for. So basically, it's just two plates, and you would inject your DNA, and then you just squeeze the tissue between there. So the issue with it, is that when you're delivering the cells, the size of the cell actually does matter. And if we think about it, cells are not necessarily round. They're various different shapes, and they usually have a long axis and a short axis. So if you can hit a cell on its long axis, you can actually use a lower voltage than if you hit it on the short axis. So it's inversely proportional to the size of the cell. So when you do it like this, really you're doing it unidirectional. And we used to think about, well, we could rotate these two plates 90 degrees and do that. And there were lots of complications with that. So we came up with this 4-plate electrode, and we call it the 4PE, which stands for the 4-plate electrode. You will find through this that we're not very imaginative in our names, but in a sense, it's, now it becomes very easy to do it in two directions. And for the skin, that's not bad because skin is pretty uniform. The cells are lined up pretty well. And we can get nice delivery. So just doing different pulse widths and applied voltages, you could see we can get different amounts of expression. So the concept was there and worked kind of nicely. But there was other issues with this. And so when you start thinking about, especially when you start looking at the potential for delivering prophylactic vaccines, it's difficult enough to get patient compliance just getting an injection. But now if you do an injection and you say you're going to apply an electric field to them, the potential to get compliance starts going down even further. So our thought was if we can make this really reduce that voltage and be able to apply it in a way that is only sort of a tingling sensation as opposed to an actual electric shock, this would work much better. So the thought was to look at the 4PE and incorporate it into a multi-electrode array, or the MIA. And this is now, the MIA is our, I guess you could say it's kind of our Italian electrode, right? It's the MIA. And, but it kind of has the same principle in that each of these is a single 4PE. So we kind of rotate the field around all this. And the nice thing about it is the gap always stays the same. So in this particular one, it's a 2-millimeter gap. Whereas with the 4PE, it was a 6-millimeter gap. So we're already able to reduce the voltage because the field is calculated on applied voltage over a distance. So the smaller the distance, the lower the voltage. And so when we do this, we can also be able to, because all these are independently addressable, we could also move the field in a way that we want. And this actually worked really, really nice. So this is in guinea pig skin, and 300 volts per centimeter is what we standardly used with the 4PE. So it was applying across that 6 millimeters, and now going across the 2, we're able to actually reduce it down to 225 volts per centimeter, and you don't really see a difference in the expression. So we could still maintain that expression. And this is typically what you would see in the skin, because you think about skin is going to turn over every 7 to 10 days or so, maybe as much as 2 weeks. So as that skin turns over, you start to lose the expression, which is fine. For many things, transient expression is okay, especially for a couple of weeks. And then the other thing that I should point out with this that we thought, that with doing it this way, we could just continually add more rows and columns and make it as big or as small as we would want. And so this is kind of what the idea here was. So could we, instead of using our standard 50 microliter injection, which makes a 6 millimeter bubble, could we do either a 200 microliter injection or 4 50 microliter injections, and then sort of apply the MIA in 4 spots, and how would that work? This is just to keep the identity of our guinea pig in there. We have a very strict IOCOC. So the nice thing about this was that when we look at this, of course, with just injection, whether it's the single 50 microliter injection at 2 micrograms per microliter, or the 4 by 100, or just a 400, you could see a nice increase with the injection only. But when we add the pulses, our expression does go up. But the really nice thing that we found was the difference between a single application versus 4 applications was only about 5-fold difference. So it was linear. So that kind of told us now if we want to get a certain amount of expression, then we know we just have to do a particular amount of it, of the area. We don't have to worry about, well, as we go up, there's a much higher level that's going on. So this was actually very encouraging when we did this. So what about for vaccines? So when we did this again in the skin, so this is injection only, and you could see that you can get some antibody production. But when we do it with the pulses, you could see that we get a much better yield of antibodies, and actually it stays elevated for quite longer. This was actually just with essentially reporter antigen. It was hepatitis B surface antigen. But this can be done. We actually did this with a protective antigen from Bacillus anthracis as well, and we also saw that work really well. We did this because we wanted to do it in guinea pig skin because that's closer to humans. So this kind of worked really well. But we still were not satisfied that we had gotten down to the best way of doing this reduction in voltage. And so we started thinking about, well, how else can we manipulate the tissue in a way that make it more susceptible to the applied field? And we thought about, what about adding a little bit of heat? And so we set up this applicator in which we had an infrared laser on one part of it in the center, and then we used 4 electrodes. So in a sense, it was just taking out a piece of the MIA, so just 4 electrodes of that MIA. And, but this time it was a 5-millimeter gap. And I don't know if I pointed out, but the MIA is set up as a non-penetrating electrode. So it just sits on the surface of the skin. And what we thought to do, so all along, gene electrotransfer has been looked at as being a non-thermal process, that heat was bad. And we were kind of going against that grain and saying, well, heat maybe is not so bad, but it has to be done in a controlled manner. So if you think about damage or pain from heat, it's temperature over time. So if you were to elevate the temperature for a prolonged period of time, you can get damage to that tissue. So we wanted to elevate the temperature to 43 degrees, but for very rapidly increase and then a decrease. And that's why we chose the infrared laser to be able to do that. And the thought was that when we did this, we would increase membrane fluidity, which is really what we want because we're affecting the membrane lipids to get this permeabilization. And then we could lower the voltage and be able to get that same expression. And amazingly enough, we were correct. So if you look at this again in guinea pig skin. So here we're looking at heat at 200 volts per centimeter, and these are 150 millisecond pulses. And you can see that we can increase it versus 200 volts per centimeter without heat. So we're about almost a log higher in expression. But to me, the more critical aspect of it, if you look at 100 volts per centimeter with heat versus 200 volts per centimeter without heat, you could see they're almost identical. So we're actually able to cut the voltage in half now, just by applying just a little bit of heat. And this was really nice. So then the next thing was, why not try and combine the positive aspects of the MIA with the positive aspects of heat? And so that's what we did here. So we took, again, our MIA array, and now what we did was we added a fiber optic in between each of these sections of 4, and we were able to split the laser into these 9 fibers. And also, as with all our stuff, each fiber was independently addressable, so we could turn it on or off. And so because we know that we could apply a voltage with heat that can deliver, but the voltage without heat would not deliver, that we could potentially start actually targeting where within the tissue we would get the delivery. So how well does this work? Actually, this works really nicely. So if we start looking at And we're actually reducing the voltage to start with, starting with what our reduced voltage was with the MIA, and we were able to do it even more. So this is the standard for the MIA right here, 45 volts, 72 pulses. And the red here is actually cutting the number of pulses in half. So maintaining the voltage, but reducing the number of pulses. And we actually had learned that as you're pulsing, I guess in public I don't want to necessarily say that we felt what it felt like, but just imagine that we did. As you're pulsing, it's actually the first few pulses you don't actually feel. It's a buildup. And I think part of it is because as you're pulsing that tissue, the impedance of that tissue is starting to drop. And as it drops, you start feeling those pulses. And it happens usually around 40 or 50 pulses. And so we thought that if we can get down below 40, that would work. And you could see that we could do that here. And we were also here able to actually reduce the voltage even further as well, which also was a good thing. And so we could reduce the voltage by an additional 23% and the pulse number by 50%. So this actually worked. We were really, really pleased with this. And so how does it work when you start actually utilizing? Oh, sorry. One other thing that we wanted to think about, and this actually goes to something that we're starting to do right now, and that's protein replacement therapy. We were thinking that for that, we would always have to use muscle. And delivery to muscle, while effective for a lot of things, is a very painful process because you think about it, you're sticking a needle in a muscle to inject the DNA, and then you're sticking needles into the muscle to pulse them. And muscles are sensitive, and so it's not a pleasant experience. So we've always been trying to do it through the skin. And interesting, when we did our standard delivery, whether it was single or multiple applications of the MIA, typically the expression was in the epithelial layer, as you can see here. And this is a single versus multiple, but still you could see it's there. But when we add the heat, we actually can get it deeper in, and actually almost down to the muscle layer without actually having to stick needles down into the muscle. So we're actually just starting now to start testing this for delivery of factor IX to see whether or not we could actually do this in a minimally invasive way to potentially treat hemophilia.
So, Richard, do you
Yeah.
think that's down to some sort of temporary disruption type junctions in the epidermis?
It's possible, and we're not sure exactly yet what's happening, but we think we are opening up pathways. And I think it's a combination of the pulses interacting with the heat, being able to do that. We also think that what's happening is, so we think even with our standard, without the heat, that the field is penetrating down, but when the field gets down low, it's much, much lower. So when heat is applied, those low fields are enough to get the delivery. And without the heat, you can't get the delivery there. And we think that's what's happening.
But what about independent of temperature effect? If you looked at individual cell membranes in terms of both fluidity and in terms of impedance, is impedance going down in individual cells separate from the junction?
So that's an excellent, we're trying to measure that now. So we're looking at it now to figure out, because we think that's exactly what's happening, is that we're changing membrane fluidity, and we're also affecting impedance. Whether it's a global tissue impedance or whether it's at the cellular level, that's what we're trying to figure out. Either would be fine because it tells us, but if it's at the cellular level, then that adds into this idea of being able to target, and that would actually be pretty good as well if we could do that. We actually have an application in right now to NIBIB to do just that. We have a colleague who's developed this way to measure impedance in real time. So we can measure it while we're pulsing. And so the thought would be is to look at all these different parameters. And then we have another way now of actually measuring membrane fluidity to be able to see how that's going. That right now we're doing in vitro. Trying to do that in vivo is a little bit more difficult, and we haven't fully figured out how to do that yet. But those are the key questions, yeah.
Those parameters reversible, come all the way back to
Yes, yes. So it is temporary unless we turn it up, and then it could be irreversible. So the next question was, that's all nice, but can it work in an applied setting? So this is, again, hepatitis B surface antigen, looking at production of antibodies. So if you remember the previous example of this I showed, that's down here. And it looked really nice when I showed you before that just with applying the fields, that we can get a nice increase over injection only for antibody production. But when we apply our heat, we were shocked that it just went through the roof, the antibody production. And we're out now, 30 weeks, and we still have high antibody levels. And so they just started to drop now. And really, this is kind of noise. If I kept this part in log, you would see that this really is very close. And so, and for now, we're going to start testing it further for actual protective studies to see whether or not this is going to actually work. But this is a key for DNA vaccines. The issue has always been, how do you deliver it in a way that you can get really good antibody production? And nobody has ever shown these kind of levels before. So we're really excited about that. The other thing that we've looked at in skin is, could we use this to deliver angiogenic factors and treat ischemic tissue? And so in this case, we use basic FGF in a rat hind limb model. This is normal perfusion of the hind limb. And then what we do is ligate out the femoral artery. That's what's happening here. And you can see that now there's very little flow through the hind limb. And then we treat either with pulses or without pulses. And within 2 weeks, you could actually see return of blood flow into the hind limb, and not when we just do the injection only. So when we started looking at this, we started thinking, well, what about doing it in the heart, for coronary artery disease? And so we thought, of course, why not, right? It's the same type of thing. You have a blockage of vessels. You have an area that becomes ischemic. You eventually get an infarct in that area. There's lots of different ways to treat it. There's ways to clear out the blockage. You could do a bypass. All of those work to a certain extent, but really the key is to try and increase the formation of collaterals, try and get more blood flow into that area that could potentially become infarcted. So our thought was, well, why not do what we were doing in the skin, do it in the heart? And I was having this discussion with a friend of mine who happened to be a surgeon who did a lot of bypasses. And I, of course, started laughing when he told me to do that, because I was like, seriously, you really want to try that? Because I would think it's not the smartest thing in the world to apply electric pulses to a heart. I mean, it's not really something that tissue likes. And so, but he said, yeah, we could do it. Don't worry. And I was like, okay. So we learned very quickly that you could do it, but if you hit the T wave with that pulse, you're going to fibrillate the heart. And so, so there's a small window that you have to be able to do it. And so we initially started with these long pulses that we were doing in the skin, and we quickly went down to 20 milliseconds in order to do that. And it actually, once you do this, it actually works really well. We were doing this in a porcine model. And as long as I had a colleague who created a software program that we could actually get the ECG into that program and monitor when we can pulse, and to make sure that we pulse just on the rise of the R wave and we're completed by the T wave. The problem was also when you do multiple pulses, whether the heart stays in rhythm when you do it. And most times it did. The problem with a porcine model is that sometimes they fibrillate just when you look at them. And so we were kind of having to balance all that. But it worked out pretty well. And I'll show you how we did this. So the first model was just normal heart, no ischemic regions, just to figure out, could we deliver? We created this electrode, which is these 4 needles, and we had an injection port in the middle. And we had to do this because unlike with the skin, we get a nice bubble that rises up and we could see what's there. In the heart, you can't see it. There's no bubble at all. So initially we were injecting and then guessing where we should stick the electrodes. And so by incorporating it all into one, we knew exactly where the DNA was and our electrodes would be there. The other thing is we had a way to control the depth of that injection needle so that we can get either into the epicardium or down into all the way down into the endocardium or anywhere in between. And so typically we would go about 5 millimeters deep with the injection, and the electrodes were 7 millimeters long is what we typically did. And then, of course, as I mentioned, we had to sync this. And then when we went into our disease model, what we would do is tie off the LAD, and we would keep it tied off. And then we would treat in 2 areas on the margin between the ischemic and non-ischemic area and in the ischemic area. The other things that we would do, since we were sort of like standing there for many hours over a pig with its heart exposed, we also were lucky to have a cath lab in our vivarium. So we actually did arteriograms before we opened them up. Then we opened them up and then used an instrument called a SPY, which took the movie about the perfusion, then tied it off, took another movie, did another arteriogram, and then treated, then closed them up. And so this usually took about 5-6 hours to do this. And I understood why I would never, ever want to be a heart surgeon because that is brutal. Anyway, so how does this work? So the first thing was, as I've been going on about, is to test different conditions and to see what kind of expression pattern we could see. And you could see, as we look at these, it's a little bit different. And the interesting thing that we saw was the pattern was different in normal healthy tissue versus ischemic tissue. And this kind of went through most of these pretty well in that it was consistent in that. So it was kind of interesting in that. And that kind of played into this idea of doing 2 in the ischemic area and 2 in the border region on that. And so these are actually some images from the SPY machine. This is a normal heart. I had the movies, but they didn't play, so I'm kind of just showing you the stills. And so you could see nice perfusion in. This is where we would tie off the LAD, and this is the treatment area. So this is after tying off. And now you could see this is all ischemic. It's a big ischemic area. We were actually afraid when this happened that the pig might not survive, but they actually did. We only had, out of about 120 pigs that we did, only one had an actual heart attack after we did this. And I'm not sure if it's because we said something or it's because of the ischemic area. But anyway, 2 weeks later, we look at this, and you could see that perfusion is actually coming back into this area. So we're actually extremely encouraged by this. And this is one of our controls. Again, you could see the perfusion, the ischemic area, and then 2 weeks later, you could see again, there's no return of that perfusion. The nice thing about the SPY, you could quantitate this as well. So at baseline, right after we tied off the LAD, you could see that the drop of perfusion was in the high 80% range. 2 weeks later, we then measured again. And in our controls, you could see a little bit of improvement. But when we get into this one condition, 20 milliseconds, 60 volts, you could see that the perfusion is almost gone, right? It's down very low. And it's about a 68% improvement. So we looked at this as being the target conditions for delivery. And then we did our long-term experiments. And so in this case, that was doing it with the arteriograms. So this is pre-occlusion. I'm not sure if you could see the vessels here in both of these cases. This sort of red dot here is indicative of where we tie off. And if you could see, there's nothing flowing past that. And then this is 7 weeks later, and this is a blow-up of this area in this square. Here's the tie-off, and you can see there's no blood flow past that. Here's our treatment, and now you could see that there's blood flow. We believe that this is a new collateral that formed, and then down here again is a new vessel that we did not find in the original one. Yeah?
Can you remind me, what's the relative timing between the ligation and the treatment delivery?
It's maybe a half hour. It's pretty quick. So however long it took us to do all the imaging and so forth, and then we treat, because we're now developing a catheter device to do it through a catheter, but at that point, because the chest was open, we wanted to just do it all at one time, because we couldn't go back in. Once we went back in, that was a terminal procedure.
So do you have any sense of how much of the effectiveness will be maintained if you start looking at longer durations to make it more applicable to?
This is the next step. So this is trying to get more funding to be able to do that. Yeah, you're absolutely right. This would be the thing. The other thing, what we're looking at too, is looking at some other molecules. And we're actually doing that in a rat model now, because it's hard to do that in a pig model. So we're kind of trying to work that out in the rat, and then we'll move back to the porcine model after that. The other thing, just to give you an idea, afterwards, and this is also at 7 weeks, you could see that in the treatment, the infarct is much, much smaller than in the injection only or the no treatment. So we are seeing a reason to do that, and it's working kind of nice in there. Okay, I'll try and move a little bit more. So I wanted to actually now just finish off with just a little bit about the cancer work that we've been doing. And for us, what we do is immunotherapy, and we call it GET IT. It's gene electrotransfer for immunotherapy. And what we target is either muscle or tumor, although more recently now we're thinking about the skin, because we've worked out a lot of stuff in the skin. And I really don't like muscle. We did muscle a lot, and I just think it has a potential for a lot of damage, and I just don't like that. And what we're looking at is plasmids encoding cytokines and delivering them either as a single agent or in combination with other agents, potentially chemotherapy or more recently checkpoint inhibitors. And again, looking at being able to get the right expression profile. So the molecule I'm going to talk about, even though we tested a lot of cytokines, I'm going to talk about interleukin-12. And interleukin-12 is a very potent immunostimulator. It's extremely potent. And when it was used as a recombinant protein a couple of decades ago, it actually ended up going on the shelf because it created such toxicity that there were even some deaths in the trials. And so even though it worked really well to control tumor growth, the problem was the toxicity on it. So we started thinking about, well, wait a minute now, if this molecule really works that well, maybe we could figure out a way to control that dose. And so we were thinking, why don't we deliver it right to where it's needed, into the tumor, and at an expression level that would give us that positive effect without the negative. So we've looked at this in a mouse model. This is a B16F10. It's a very aggressive melanoma model. We looked at 2 different plasmids, one just a standard run-of-the-mill single promoter mouse IL-12 plasmid versus an optimized dual promoter IL-12, a couple of different GET protocols, a couple of different arrays, and then looked to see what was going on. So we were able to get some different expression profiles. This is what we call EP1, which is 100 microseconds, 1,300 volts per centimeter, with a needle array. And this is 6 needles in a circular array, and the pulses rotate around in a 360 degrees, 1 pulse per 60 degrees. And versus longer pulses, 5 milliseconds or even 100 milliseconds, at lower voltages with caliper electrodes. If you remember, I showed you a picture of them, that just 2 plates. With the non-optimized or the optimized, or doing EP1, EP2, but reversing the electrodes. And it's interesting patterns that you get. So this one, EP1, you could see is sort of intermediate, right? Here's injection only, and here's these guys, high expressors, right? A log order higher than here. With the optimized plasmid, interestingly enough, EP1 starts getting a little bit closer to these guys, although these guys got a little bit higher as well, but so did this. And then finally, when we switched the arrays, the electrode arrays, it was interesting that these two almost became equivalent, which was kind of an interesting observation. So it shows you not only the parameters count, but the electrodes do as well. So what about response? So when we look at these and just look at this treated tumor, everything works really well, right? So EP1 with the 6 needle, we got 10 out of 10. With the caliper, was 7 out of 9. The optimized, we had 8 out of 9. Same thing with EP2, 8 out of 10 with the caliper, 10 out of 10. Interesting, with the optimized one, where we got that really high expression, only 3 of the treated tumors went away, which we had never seen before. But what we really wanted to know, what happens when we now challenge them? Are we actually getting an immune response? And that's where it becomes interesting. The difference between the expression levels. So if you remember, this is actually an intermediate expression, and this actually gave us our best immune response. So even here, when we switched and just elevated a little bit, it dropped the protection. And here, where we got our really highest expression, we saw virtually no protection at all. So this kind of told us that we need to actually understand how much expression we're getting. So after doing a lot more tox studies, biodistribution studies, we were able to convince the FDA to allow us to go into the clinic. And this was actually the first time that gene electrotransfer was used in the clinic, and this is why it took us about 18 months of negotiations with the FDA to do it. And this was done in late-stage melanoma patients. We were able to, we were allowed to treat between 2 to 4 lesions. There were 5 dose cohorts escalating, and this actually ended up being 1.6 mg per ml just because the manufacturer actually didn't get up to 2 mg. And the dose was 25% of the tumor volume. So if it was a 1 cubic centimeter tumor, the dose was 250 microliters. And the maximum that could be injected was 2 mils. So I will tell you, at the end of doing these 5 cohorts, there was no toxicity found. So in typical fashion, the medical oncologist said, we have to up the dose because there's no toxicity, and we have to keep going to this toxicity. And so we added 2 more dose cohorts at 2 mgs and 3 mgs and gave them the full injection volume of 2 mils. And we did similar to the mouse study where they were treated on days 1, 5, and 8, 3 times over 1 week with 100 microseconds, 1,300 volts per centimeter. So it actually worked really well. So there was no toxicity seen. Locally, it was very effective. But the thing that we were really surprised to see is that in 3 patients, one of them who had another treatment after the IL-12, all their cancer went away. And I'm going to show you a picture of one of these, and they have stayed away for years. They have not recurred. And we did not expect that. We thought we could get a vaccine effect, maybe protection, stop new tumors from forming, which we did see that in 7 other patients. But this is an example. This is one of our patients. All this is melanoma, front and back. This is 3 of the 4 lesions that were treated. This is about 10 months later, and he looks stable, and that's what we thought. But really, when you start looking at these clusters, you could see that these tumors are collapsing, they're shrinking, and 18 months later, you could see that they're all disappearing. And at this point, they wanted to put them on another study, and so they needed documented evidence of disease. So they started biopsying these lesions, and they all came back negative. And they basically, the pigment, what they are macrophages picking up the released pigment from the dying melanoma cells, and these are called melanophages. But it actually, he's out now, I think 10, 11 years with no disease. There was another patient that made it 7 years. She passed away, but from natural causes, not from cancer. And the 3rd patient we lost track of. He was happy, and he didn't want to come back to the cancer center. And so we don't know where he went. But it's interesting because a good thing and a bad thing. So when we started the trial, we were worried that we would see nothing, which happens quite often when you go from a mouse to a human. And so one of the clinicians suggested, well, why don't we, since we're doing 4 lesions, why don't we biopsy 1 at 11 days, 1 at 21, And then the FDA required us to take out all the treatment areas by 42 days. And so we said, okay, if we're doing that, we could do that, and then we can measure whether or not we actually have IL-12 being produced. Because the worry was, if it didn't work, is it because we didn't get expression, or it's because it doesn't work? And so, interestingly enough, we start seeing a pattern. So this cohort, which is the 0.5 mg per mil, 2 of the 3 patients that had the complete responses were in this cohort. And you could see their expression, as well as patient 14 here, are in this intermediate moderate. Our stable and partial are actually in between very high expression and this sort of intermediate. And the really high expressors ended up being progressive. And it was really interesting because, I don't know, it was really shocking because it actually correlated with our mouse studies, right? The intermediate expression gave us good results, actually complete responses. And then a little bit higher, but not too high, gave us stable disease or partial responses. And the very high levels gave us progressive disease. It didn't really work. It worked locally, just like we saw with the mice, but we did not get the systemic expression or the systemic response that we wanted. So this went to phase 2. And again, in this case, in the phase 1, there was only 1 round of treatment. In the phase 2, the clinicians said they wanted to do multiple rounds, and it actually worked well again. So, oh, sorry, this is actually not the right slide on there. Here it is. So I'll have to go back to that one. But in the phase 2, we actually ended up seeing complete responses in about 15% of the patients. 4 of the 29 got this complete response. And responses in at least 1 untreated lesion was almost 50%. So seeing that distant response. So again, this kind of told us that this is something that could be really good. Again, looking at biopsies, we started looking at why are we missing in some of these other patients? Why are some doing so well and others are not? And so we were thinking it could be due to expression, but what we were turning out that those high expression ones, what was happening was the levels of PD-1 and PD-L1 were being elevated. And so this was blocking the immune response. The other thing that we started thinking about was in about 65% of melanoma patients, they don't respond to anti-PD-1 or anti-PD-L1 because their tumors are what's called cold. They don't have lymphocytic infiltrate, and they have very poor expression of PD-1 and PD-L1. So the thinking was, well, since we're elevating them, what if we added a checkpoint inhibitor? Maybe this would now take care of those patients that were not responding. So we kind of did a quick mouse study where we did 2 tumors, and then we treated 1 tumor, and then we combined it with a checkpoint inhibitor. And we found that when we added the anti-PD-1, we actually got a much slower growth of that second tumor. And so this then went into the clinic, looking at patients that had gotten the IL-12 and then either went directly into the checkpoint inhibitor therapy or had something in between and then went to checkpoint inhibitor therapy, and then looked at their responses. And what we saw was amazing. So 4 out of 8 of the patients had a complete response. So their disease completely went away. 2 others had partial, 1 was stable. But 7 out of 8 patients had a response that was systemic. With the intervening therapy, interestingly enough, this dropped a little bit, but it was 65%, 66%, still pretty good. So this led to a phase 2 of doing this in a prospective way, of really doing a combination. So doing the IL-12 delivery together with Pembro, and then in the next 21 days, they just got the checkpoint inhibitor and then repeated the cycle again as long as they could. This was done at UCSF and at the University of Utah, and Oncosec Medical was the sponsor of this. And this trial ended about a year or so ago. And again, really, really amazing results. 22 patients. At 21 months, 11 of these 22 patients had responded. When they went longer, it ended up actually going up to 59%. The complete responses at the time, at the stopping point, which I think was 6 months, was 9 out of 22. This is now elevated to 11 of the 22, and all 11 are still 100% out now. I think 25 months, none have new disease at all. So very, very impressive on this. So this has now moved into a registration trial with the FDA. It's been given fast track status, and the hope is the end of 2019 or early 2020, this combination could get approval. And now it's being looked at other indications as breast cancer and some Merkel cell, squamous cell, looking at the potential for lung and pancreatic as well. So in summary, hopefully I convinced you that electrotransfer is really a nice tool to use for being able to deliver DNA and in a controlled manner. I showed you some examples on ischemia and cancer as well as DNA vaccines. I didn't show you our wound healing data, but that works really well also. It's really important. I know I've been saying this over and over, but the expression pattern is very, very critical. I think this is really what separates the acceptance of gene therapy versus protein therapy, because the FDA and clinicians know with protein what they're putting in. With DNA, of course, you're putting the DNA in. The question is how much protein is coming out. So I think the closer we get to, in gene delivery, to actually being able to predict how much of that effector protein is going to be produced, the easier it will be to get approval from the FDA. And then for the get it, the key was getting the right cytokine expression in the right place at the right time. I had some additional data that showed why the electrodes ended up being so important. It's because where the expression of the IL-12 was, and that kind of gets back to some of the targeting that I talked about earlier. So just kind of with respect to that, with the cancer, monotherapy versus the combination, same thing again, be able to get that right expression, be able to see that significant increase in tumor-infiltrating lymphocytes, get the right memory response, and get that effect that you're looking for. In a combination, it's the same thing. We want to take this, but then either put checkpoint inhibitors or other cytokines or other factors that actually can boost it even further. So finally, I just want to thank all the people that have been involved in all this work, all the collaborators, and all the current and past members of the lab, and then our funding. And thank you for your attention and also the great questions that you asked already. Yeah. I think he outranks you, so. Go ahead. Good.
Hi, good time. So with respect to the heat delivery in addition, you didn't do that with like the ischemia model or the cancer model, but I saw like the voltage increase. So what was the deciding factor for that?
That we did all that before we started playing with the heat.
Oh,
so,
okay.
but it's actually a really good point, which I didn't mention. So kind of that stuff that I was talking about early on, especially with heat, this is like all within like the last 2-3 years that we've been really doing this and really working it out. So the big step was the transferring it from just a standard 4-pin electrode to the MIA. So now we're looking at ways, okay, now how do we take that? We think we could pretty much do like the melanoma tumors, and we've now designed an electrode to be able to do that. And I think with the peripheral ischemia, I think we could do that as well, and I think we'll try that. With the heart, we have to start thinking about that, how we would do it, and we could potentially take one of the fibers and put it into the catheter and be able to do that. But right now, it's a matter of logistics because we have wires, we have an injection, and now we would have a fiber. So that just is more complication, but it's just something that you have a problem, you have to find a solution to it in there.
this question, I want to preface it by saying I am a dyed-in-the-wool experimentalist. I believe tremendously in the experimental approach. You have done heroic things with parameters and doing all this, but it seems to me this is also an area that's ripe for computational approaches. And I'm just curious, one could model the tissue parameters. We talked a little bit about, membrane input and resistance.
Absolutely.
Anyway, I'm just curious, in terms of coming up with all kinds of interesting combination of parameters, is there a parallel computational
So
approach to that?
there is to a certain extent, and we tried that many years ago because we also were tired of having to do 50 combinations of parameters to figure out what we're doing and to do it. And we've gotten better at that. And we started trying to do some modeling and some, like a simplex array and some computational work. And it just never matched. And it was kind of strange. And I think now we might have I think it was a matter of not enough data. And I think now we have, because we're accumulating so much data now, that it might work a little bit better, especially now that we understand a little bit more of the fluidity aspect and the conductance aspect and how the electrodes play in that we might be able to do a little bit better with that. I think the biggest problem that we're having is, and this is how I mentioned before, I'm sorry, I'm kind of going all over, but with the colleague of mine that now we could measure impedance in real time. I think that will be the key piece, because what always didn't match was, when you're doing the computation, you're assuming homogeneous conductance and impedance within that tissue, and that's far from reality. I mean, depending on how you're doing it, and if you're sticking needles in, you've now changed everything now, because you have these conductive sticks in there, and you've also made these pathways through. I mean, you're penetrating in. So skin might be a good model for that. But again, as you go through the different layers of skin, the stratum corneum, then you get into the dermis and the subdermis and the fat, all of those have different properties. And so the models are getting better, but so far we've not had luck. And if anybody has any ideas how to do it, I would love to hear it, because it would make our life a lot easier to do that. And I think it might take another couple of years to really build those models where you could do it. But eventually, I think our software and our understanding of the tissue dynamics will get better, and we'll be able to do that.
Just one follow-up question. You mentioned fluidity several times. So it's pretty well known that dietary changes in relative degree of saturated lipids can change membrane fluidity. I think of the examples of hibernating animals. They're really amazing, like the 13-line ground squirrel. You feed them these different levels of hydrogenated lipids, and the suppleness of the whole body and the fluidity of the membranes is different. Might you be able to manipulate dietarily the fluidity?
It's possible. It's possible. The question would be is, can you get it in the right area? And is it going to be uniform everywhere? So we've actually tried this in diabetic rats as well. So much higher fat contact, these are going to be obese, and there are changes. It is different than when you do the non-obese rat versus the obese rat. So I think there's something to look at with that. But I think until we get to a point where, we could be confident that that would affect globally, I think. But if we could certainly find, if there was a way to actually pre-test and say, because particularly for the skin, if you're delivering to the skin, you could deliver anywhere. It doesn't matter where in the skin you deliver, unless there's a specific, maybe if it's psoriasis or something that you have to go to a particular area. But if it's for a vaccine or something like that, it shouldn't matter. It might be a way to test, okay, what's the content there and so forth, and how would you do it? And if we can come up with a quick test to do that, that might be a way to do that.
You're coming here.
Okay, so to what do you owe the response of distant lesions to a localized treatment?
Yeah.
Is that the solubility of the protein? once it's
No, actually, it's cellular. So, the interesting thing is that the IL-12 after a few days is out of the picture, right? So, basically, it's an ignition switch. So that's the nice thing about 12. It's a very potent stimulator, and it starts a process. And actually, I didn't show this, but one of the first steps it does is it initiates the production and release of interferon gamma, from nearby dendritic cells and macrophages. And gamma is actually the effector molecule that you want. And so gamma starts this cascade. In addition, the second thing that the IL-12 does is that it will get infiltrate to come in. So I mentioned about lymphocytic infiltrate. So mainly T cells would come in. And I kind of alluded to the idea of where the 12 is. And what we found was that when you have the 12 being produced more in the periphery of the tumor, and now the infiltrators coming in, and as they come in, they're bathed in the IL-12, and they're upregulated and activated. And then they come in, and gamma is within the tumor, and all this starts this environment to train these T cells that this tumor shouldn't be here, kill that tumor, and then they go back out into the lymph nodes, and the clones expand, and then they go out. This is why it takes so long, because it's a long process. That's why in some of the patients, we just thought, okay, this is great. We stabilized them. We're going to give them more time. They won't have toxicity, and it'll be a better life for them. But then after 6 months, we started seeing the tumor starting melting. And then after a year, they started getting even further away. And it just takes time for these cells to come in and just chew away these tumors. And that's what it is. And so the 12 is kind of just that starter molecule. And so that's why they started looking at doing it in repeat cycles, because, okay, we got it going. Now let's just reinvigorate it again. And that seems to work pretty well in the humans.
Yeah, in answering some of Rachel's question, and where she was talking about the difference with heat and voltage, you were talking about it's difficult to apply heat to certain places in the body. Have you considered looking at the technology kind of associated with focused ultrasounds and moving that way for heat penetrance rather than traditional heat?
Yeah. It's a thing that we've looked at. We actually know a few people that actually are doing sonoporation, where they're doing delivery with. And there are good things and bad things to that. So again, it's a little harder to target. They're trying now with the microbubbles to be able to do that. We've thought about it, and we might try it. The other thing is to do it not necessarily to do the ultrasound for the delivery, but we could also generate heat with the ultrasound, which I think is what you're alluding to. But we could also potentially do it with microwaves. And so the question is, which of these could work without adding any potential damage or discomfort? And again, it's this thing about temperature over time. So how fast will it rise, and how fast will it dissipate? And so at 40, we picked 43 because 43 is relatively safe for about 10 minutes or so. Once you start getting past that, you get to 15 minutes, you could start sensing the heat, and you get more towards 20, you could start getting closer to burns. At 45, interesting, is really the cut when that time limit really gets short, and you're right at 3 to 5 minutes where you could start getting burns. And at 50 degrees, you're in the seconds. I mean, it's kind of like you touch something hot, and you're just going to go right back out of it. So that's the balance right now. The other thing we thought about was, instead of using infrared, was to use LEDs. And that we could have better control over. We could actually incorporate those a little better into the electrodes and put them in and just have the light come out of the electrodes. So we're thinking about all these. It's kind of like I was saying, it's daunting as you add all these variables in, but it's fun. It gives us something to do, keeps us out of trouble. You know?
So the IL-12 data is really interesting. It kind of taught, I think what it taught me was that you've got to hit that sweet spot with just a very high degree of precision. So you must be thinking about other ways of keeping it within that sweet spot.
Yeah.
So you know, what do you think is worth going after? Like promoters that you can sort of regulate the gene dosage, this sort of thing?
Yeah, we've given this a lot of thought, because, you know, in a mouse, it's a lot easier to control it than it is in a human, because in a mouse, we know exactly, we're putting that tumor in the exact same spot every time, as opposed in a human, it could be anywhere. It could be in the arms, could be the legs, could be in the trunk, could be on the head, anywhere. And so in each one of these tissues is different, even though It's still within the skin, but there's different properties within it. So we thought about that, and we actually had tried an inducible promoter, and it really doesn't work as well because most of them are leaky, and so you're still going to get this constitutive expression. And with 12 especially, having that extra 12 is not good, and it leads to toxicity. The other thing we thought about was, could we somehow monitor this and find something out of the circulation that will tell us that it was delivered properly and the right cascade has started? And so we've been looking at that, and we think we found possibly something that could tell us that. But in the interim, we had the combination with the checkpoint inhibitors came out, and it elevated the response rate so much that the idea was, well, maybe we should focus on that and not necessarily the monotherapy. Although I think the monotherapy still has a lot of advantages. Mainly, you don't have the expense of the checkpoint inhibitors, which are hugely expensive. So it is something that we're still looking at. And we haven't quite figured it out yet. But we hope at some point we may come up with something that could work.